Dr. Rajendran's Institute of Medical Education. Free PG Medical Entrance Coaching. Get more MCQs explained with figures and picture quizzes for PG Medical Entrance free to your mobile. Send WhatsApp message YTMCQ to +917558877764.
This video about the actions of insulin and about insulin receptor is meant for undergraduate medical students. The main site of action of insulin is the liver, but muscles and adipose tissue are also important. This figure shows the most important actions of insulin. The actions of insulin on carbohydrate metabolism, on protein metabolism, and on fat metabolism. The blue lines indicate the steps which are stimulated by insulin, and the white lines indicate steps that are inhibited by insulin. Insulin stimulates the conversion of glucose to glycogen that can be stored in the liver and also in the muscle. Equally important, insulin inhibits the conversion of glycogen back into glucose. Following a meal, the glucose tends to rise in the blood. Also, the presence of food in the gastrointestinal tract stimulates the secretion of insulin. So, rising glucose in the blood and a variety of hormones secreted by the gastrointestinal tract stimulate the secretion of insulin following a meal. The high insulin levels following a meal stimulate the conversion of glucose to glycogen, and this glycogen gets deposited in the liver as well as muscle. Now, in between meals, that is, for example, at midnight, the glucose tends to fall. The low levels of glucose removes the stimulus for insulin secretion, and the insulin level in the blood is very low. The low levels of insulin in the blood removes the inhibitory effect on glycogen getting converted back into glucose. In other words, when the levels of insulin are low in the fasting state, glycogen gets converted to glucose and the blood glucose level tends to be maintained. It doesn't fall. In short, following a meal, when glucose is available in plenty, insulin stimulates the conversion of glucose to glycogen and that gets stored in the liver. In between meals, that is during fasting, insulin permits the conversion of glycogen back into glucose and the blood glucose level is maintained. This is very important because glucose is the only fuel that brain can utilize. So even during fasting, blood glucose level is maintained by the low levels of insulin and the brain is fed with adequate levels of glucose for its metabolic needs. Now let us see what are the effects of insulin on fat metabolism. Now following a meal, when glucose is available in plenty, it gets converted to glycogen and is stored in the liver. When the liver is saturated with the glycogen, the excess glucose available gets converted to fatty acids, and these fatty acids are converted to triglycerides, which is the storage form of fat, and it is stored in the adipose tissue. The conversion of glucose to fatty acids and the conversion of fatty acids to triglycerides so that they can be stored in adipose tissue, both these steps are stimulated by insulin. Importantly, the conversion of triglycerides back into fatty acids is inhibited by insulin. In short, insulin stimulates lipogenesis and inhibits lipolysis. What is the effect of insulin on protein metabolism? Insulin stimulates the conversion of amino acids to protein, and insulin inhibits the conversion of protein back into amino acids. An important substrate for making glucose is amino acids. Insulin inhibits the conversion of amino acids into glucose. The steps that are stimulated by insulin, that is the conversion of glucose to glycogen, the conversion of glucose to fatty acids and then to triglycerides, and the conversion of amino acids to protein. All these are steps which are stimulated by insulin. These steps are increased by feeding. That is, feeding stimulates insulin release, and these steps go in this direction. The steps that are inhibited by insulin, that is the conversion of glycogen back into glucose, glycogenolysis, or the conversion of triglycerides back into fatty acids, that is lipolysis, or conversion of protein to amino acid and then back to glucose. All these steps which are inhibited by insulin are increased during the fasting state. During fasting state, the levels of insulin are low. So the inhibitory effect of insulin on these steps are removed and the reaction goes in this direction. "90% of patients with diabetes have type 2 diabetes. In type 2 diabetes, there is resistance to the actions of insulin. That is, there is reduced conversion of glucose to glycogen and there is accelerated conversion of glycogen to glucose, leading to hyperglycemia. There is reduced conversion of fatty acids to triglycerides and there is accelerated conversion of triglycerides to fatty acid, leading to high levels of fatty acids in the blood. This leads to accelerated atherosclerosis and a much higher incidence of myocardial infarction and cerebrovascular death. This figure summarizes the action of insulin. What are the actions of insulin on the liver? Insulin inhibits gluconeogenesis, that is the synthesis of new glucose. Insulin stimulates glycogen synthesis, that is glycogenesis. And insulin stimulates lipogenesis, that is synthesis of fat. What are the actions of insulin on adipose tissue? Most important, insulin inhibits lipolysis. Even very low levels of insulin can inhibit lipolysis. And insulin stimulates glucose uptake by the adipose tissue and fat synthesis by the adipose tissue. What are the actions of insulin on striated muscle? Insulin increases glucose uptake by the muscle and it also increases synthesis of glycogen and protein by the muscle. Thus, the main insulin actions. Insulin stimulates glycogenesis and inhibits glycogenolysis. Insulin stimulates lipogenesis and inhibits lipolysis. Insulin stimulates protein synthesis and inhibits protein breakdown. I have told you the main site of action of insulin is the liver. In the liver, insulin decreases glucose output by inhibiting gluconeogenesis and stimulating glycogen synthesis. Insulin also inhibits the formation of ketone bodies by the liver. In diabetes, where there is deficient insulin action, there is increased glucose output from the liver, leading to hyperglycemia. Also, in diabetes mellitus, there is a tendency for ketogenesis. In muscle and adipose tissue, insulin markedly increases the uptake of glucose. These actions of insulin are brought about by the binding of insulin to insulin receptors. Insulin receptors are present on virtually all cells of the body, but the highest concentration of insulin receptors are found in the liver, in the muscle, and in adipose tissue. Insulin receptor belongs to tyrosine kinase class. This figure shows the insulin receptor. This is the cytoplasm of the cell, and this is the plasma membrane, and this is the insulin receptor. The insulin receptor has two alpha subunits and two beta subunits. The alpha subunits are entirely outside the cell membrane. This is the cell membrane, and the alpha subunit has the binding site for insulin. The beta subunit penetrates through the cell membrane into the cytoplasm. Disulfide linkages bind the alpha subunit with the beta subunits. They also bind the alpha subunits together. The end of the beta subunit projecting into the cytoplasm of the cell has tyrosine kinase domains. Insulin can bind to the alpha subunit. Once insulin is bound to the alpha subunit, the beta subunit gets automatically phosphorylated, activating the tyrosine kinase enzyme. The alpha subunit normally has an inhibitory action on the tyrosine kinase activity of the beta subunit. The binding of insulin to alpha subunit removes this inhibition, and the tyrosine kinase enzyme gets activated. The activation of the beta subunit by phosphorylation results in the activation of two other pathways inside the cell. First is phosphatidyl inositol signaling pathway. Activation of this pathway by the binding of insulin to the alpha subunit results in the entry of glucose from the blood into the cytoplasm through specific glucose transporters. Phosphatidyl inositol pathway is also involved in the synthesis of lipids, proteins, and glycogen. This pathway is also involved in cell survival and proliferation. The second pathway activated by beta subunit phosphorylation is mitogen activated phosphokinase signaling pathway. This is involved in cell growth, proliferation, and gene expression. In short, when the insulin binds with the alpha subunit, the beta subunit gets activated, causing activation of the phosphatidyl inositol pathway, causing entry of glucose from the blood into the cytoplasm. The whole sequence of events occurs within seconds. So, glucose transporter 4 is the glucose transporter stimulated by insulin, and it is present in the muscle and adipose tissue. An important effect of activation of the phosphoinositol signaling pathway is translocation of glucose transporter present in the cytoplasm to the cell membrane so that glucose can enter from the blood into the cell. The glucose transporter 4 is present mainly in the muscle. it is present in the cytoplasm of the cell. Within seconds of insulin binding to the alpha subunit, the glucose transporter in the muscle cell is translocated to the muscle cell membrane so that glucose from the blood can enter the inside of the muscle cell. This happens very quickly. The half-life of insulin is just 5 minutes, whereas the half-life of insulin receptor is 7 to 12 hours. The two major tissues containing glucose transporter 4, which is sensitive to insulin, are 1. Skeletal muscle and 2. Adipose tissue. Within seconds of insulin binding with the alpha subunit of the insulin receptor, glucose transporter 4 present in the cytoplasm of the muscle cell is translocated to the cell membrane and glucose rapidly enters from the blood into the inside of the muscle cell. When insulin is no longer available to bind with the alpha subunit, the glucose transporter in the plasma membrane or the cell membrane of the muscle cell is translocated back into the cytoplasm so that no more glucose can enter from the blood into the cytoplasm of the muscle cell. So, glucose transporter 4, which is sequestrated mainly within the intracellular compartment, that is the cytoplasm of the muscle and adipose cell, does not function as a glucose transporter until insulin binding and signaling causes translocation of the glucose transporter 4 to the muscle cell membrane or the adipose cell membrane. There is a tendency for the blood glucose to rise after a high carbohydrate meal. This tendency is prevented by insulin by facilitating the entry of glucose into skeletal muscle and adipose tissue. Insulin causes the disposal of glucose after a meal by causing the entry of glucose into skeletal muscle and adipose tissue. We already seen within seconds of insulin binding with the alpha subunit, the glucose transporter 4 present in the skeletal muscle and adipose tissue are transported to the plasma membrane, facilitating the entry of glucose from the blood into the muscle cell and adipose tissue, preventing a rise in blood glucose level after a carbohydrate meal. So normally the muscle is not permeable to glucose unless insulin is present in sufficient quantities. But exercise can cause glucose transporter 4 translocation to the muscle cell surface by activating mitogen associated protein kinase pathway. So when you are exercising, even without the presence of insulin, the muscle becomes permeable to glucose. Moreover, exercise increases insulin sensitivity of muscle, and this increased sensitivity persists for several hours after exercise. Regular exercise can cause prolonged increase in insulin sensitivity. This is very helpful in the diabetic patient. The number of insulin receptors per cell decrease on exposure to increased levels of insulin. High levels of insulin can occur in diabetes mellitus type 2, obesity, and in acromegaly. Insulin is secreted only by the pancreas. A glucose concentration more than 70 milligram percentage stimulates insulin synthesis and insulin secretion from the pancreas. Following food intake, when the blood glucose starts to rise, insulin secretion is stimulated. This is the pancreas. The veins that drain the pancreas empty into the splenic vein. The splenic vein joins the superior mesenteric vein to form the portal vein, which is the major blood supply to the liver. This is a very convenient arrangement in that the nutrients, that is the carbohydrates, proteins and fat absorbed from the diet enters the inferior mesenteric vein and superior mesenteric vein and ultimately reach the liver through the portal vein. The insulin secreted by the beta cells of the pancreas also enters the splenic vein and reaches the liver through the portal vein. Thus, insulin can take care of the nutrients, that is the carbohydrates, the fats and proteins absorbed from the diet. Insulin is an anabolic hormone. Insulin helps in the storage of the carbohydrates, fats and proteins absorbed from the diet. Insulin promotes energy storage. Insulin helps in the conversion of substrates into glycogen, triglycerides and proteins. That is, the carbohydrates are converted to glycogen and stored as glycogen in the liver and muscle. The fats absorbed from the diet are stored as triglycerides in the adipose tissue. The amino acids absorbed from the diet are stored as muscle proteins. Following food intake, the carbohydrates that are absorbed are used for the body's metabolic needs. Carbohydrates in excess of this is stored as glycogen in the liver and muscles. This storage of glycogen in the liver and muscles is favored by insulin. There is a limit to the capacity of the liver and muscles to store glycogen. Excess carbohydrates that cannot be stored as glycogen is converted into fats and stored in the adipose tissue. This conversion is also facilitated by insulin.
Get more MCQs explained with figures and picture quizzes for PG Medical entrance free to your mobile. Send WhatsApp message YTMCQ to +9175588777764.